Right? So the last class section, so we started the disease cycle. So in the disease cycle, right? So once the pathogen inoculum received to the host, it may be the primary inoculum or it may be the secondary inoculum. So thereafter, so the pathogen should penetrate, right? So there are several ways the different pathogens, they have their own mechanism. For example, last class I discussed with the fungi. So they are producing the penetration peck for the direct penetration. And also if there is a wound or some of the insect creating damages, then act as a foci for the penetration of this pathogen. So for the bacteria, it needs wounds for the penetration. For the viruses, so insects are helping, right? Vectors are helping to virus to go in. So for the broadly, there are three ways of the pathogen in penetrations, right? So the initial penetration, so initial invasion, sometimes it's called as an invasion. Once it penetrated, then going in is called as an invasion. Invasion of the host by a pathogen may be in three ways. Direct penetration, so penetration through natural openings, especially the bacteria and some of the fungi, they are choosing these natural openings. And penetration through wounds, so mainly all the pathogens, they can go in if there is a wound, right? If there is a wound. So the direct penetration, so for the direct penetration, so fungi, they are producing host story. I told you. So when the spores is reach the host surfaces, it should stick with the host. Thereafter, it will produce some germ tube. So at the end of the germ tube, we call it as a penetration peck, and the another organ we call it as a host story. Right? The host story can direct, right? The hostonia can penetrate directly, or the penetration peck can penetrate directly. Right? So penetration structure, it may be hostonia. This is, you would have seen that one in the halotropics, in the parasitic plant. Halotropics, would have seen in the onion field, and other one is there, Loranthus. Sometimes I don't know. In the mango trees, would have seen that one, right? We call it as a in Tamil Kuriviche, right? So Cascuta, right? Cascuta and the Loranthus, two high parasitic plants, they don't have any roots. So they are producing hostoria directly into the trees. From that, they are drawing nutrition. And also they can produce direct subcuticular only. So this hostoria cannot go in. It can penetrate only the epidermis, external host surfaces, and it formed inside the host. So direct intercellular mycelium, this direct intercellular mycelium go deeply in between these host, in between the host cells, or it can penetrate directly the host cells, and it can form a structure we call it as a hostoria to draw the nutrition. Or either it can produce hostoria directly, or the subcuticular mycelium can produce hostoria, right? Hostoria. So at the end of the end of the hostoria, so once this hostoria is produced, then thereafter mycelium will further advance and it will produce another structure we call it as an abrasorium, right? Abrasorium, penetration peck, intercellular mycelia. These are three structures. You can see that. I will show you the one picture is there. I have put it here. Right. So the spores, this is a germ tube, right? Here you can easily find it. I can show you the highlights. It's a spore of these fungi. It's a germ tube, right? So in the germ tube, while they are producing, directly it can form host aprosoria. This is we call it as an aprosoria. Oh, it can going while going, it can produce. Or sometimes you see, I can show you here. This penetration peck directly penetrate. Thereafter, it produces so much of subcuticular mycelium. From this mycelium, it can grow through this host here, through these host cells. Then it can produce a hostoria to draw the nutrients. Right? So this is the approach here. This is the aprosoria. You can see this is the aprosoria here. Aprosoria. This is the penetration peck. So this is intercellular mycelium. This is an intracellular mycelium. So they can produce, and at the end they can produce some of the structure we call it as a hostoria to draw the nutrition. So don't forget, this is an aprosorium. This is hostoria. This is intracellular mycelium. Then it's an intracellular mycelium. So the way, right? In this way, so they can go and they can draw the nutritions. So common type of penetration by the fungi and the nematodes, the only type of parasitic plants. They are the only type of parasitic plant. That's a direct penetration. So fungi, nematodes, they are having the stylet. Fungi, they are having the aprosorium or penetration peck. And parasitic plant, I told you, they have these direct hostoria, right? Direct aprosorium and then direct hostoria to draw the nutritions. For the fungi, it need a fine hypha produced as, by the spores. That's what I told you, the penetration peck. So then it is producing the aprosorium. Right? Penetration peck produced by an aprosorium. That's what I drawn that picture, right? Penetration. So this is formed at the point of contact with the surfaces. Contact with the surfaces. So penetration is through the mechanical forces, so softening of the cell wall by an enzyme. Right? So for example, I am going again. This is a spores. This is a spores producing first tube we call it as a germ tube. So once it is reached, this picture is very clear, right? Germ tube. So at the end of the, so once it germ tube growing, then if it is reached the host surfaces, this produces a structure we call it as an aprosoria. This is a broad structure as act as a suction pad to trap the host. So at the end of this aprosorium, it producing a penetration peck. It's a very sharp needle structure. So aprosoria is producing so many amount of enzymes. These so many enzymes may be pectinases, cellulases, or semipectinases. These can degrade this area. Then the pushing of this aprosorium with penetration peck, immediately this mycelium go in. Thereafter, this is an invading mycelium. This is an invading intercellular or intracellular mycelium. I think you got it, right? Intracellular mycelium. The parasitic plants also using the same way to penetration. Penetration peck emerging from the aprosorium. Peck grows, fine hypha reaches the normal diameter. Once it's there, it is inside the cell. That's what invading mycelium. Right? In the case of apple scab, for example, some pathogens are example, right? For apple scab, the fungus penetrates only the cuticle and stay between the cuticle and the cell wall. This is the structure here, apple scab. There is no any further invasion here. This mycelia produce in between the cells and the cell wall, so they are proliferating. They are proliferating. From that, they will produce directly aprosoria to grow the nutrition. There is no any going inside. Right? Going inside. The parasitic plants also penetrate in that way. That's what I told you. The Loranthus and Kescutas also, they are producing directly aprosoria to draw the nutritions. Here, this is a hostoria. This is an aprosoria. This is a germ tube. It's an invading mycelium. Initially, this invading mycelium is very narrow. In this area, that's what we call it as a penetration peck, very sharp needle-like structure of the mycelium, very constricted, very narrow, right? Narrow. So here, this is the steps. This picture is showing that once it's reached the spore to the host surfaces, it's produced germ tube. So at the end of the germ tube, it's produced Aprosoria A. So at the end of the Aprosoria, it's producing the very delicate, sharp needle-like mycelium we call it as a penetration peg. Then this penetration peg widened and further invading, right, intercellular mycelium, IM, right, intercellular mycelium. So then it can go maybe intracellular. So this is the way the fungal is once it's reached the host surfaces, if it is penetrated, right? So penetration through natural opening, second steps. So there are stomata and lenticle and hydnothodes. These are the natural openings present in the plant. You would have studied in the plant physiology. In these openings also, some of these bacterial cells can choose if some of the fungal spores, especially the rust, basidiomycota fungi, rust fungi, they are penetrating through the stomata. Majority of these bacteria passing into the host cells through this natural opening, 3 ways of natural opening. Right? The stomata, it's going in. So then penetration through wounds. So maybe wind or other animals or insects, or that while we are the human being doing the intercultural population can create the wounds. If the wound is created, any type of organism can go in. Right? Any types of organism can go in. So for these wounds, all type of organism is coming under this category. Right? Coming under this category. Then invasion. I told you about fungal. It is there. So then, fungal host should the pathogen should recognize the host. Host recognition is we call it as sometimes infection. Right? So the host recognition is successful, then it can able to grow further and draw the nutrients. Right? It can go further and to draw nutrition further. So the incubation period, the time interval between the inoculation to the symptom development, I told you this is we call it as an incubation period. And latent infection is there, state in which a host is infected by the pathogen, but the symptom does not show, the pathogen does not show any symptoms. That is, we call it as a latent infection. That's, for example, the COVID-19, some people are showing symptoms. Some people are not showing the symptoms because it is there as a latent infection. These people can spread quickly to the others. Right? Like that. So in some hosts, it will not show the symptoms. This we call it as a latent infection. Then growth and reproduction. Pathogen will grow and multiply within the infected host. So as a fungus, as a spore, bacteria as a replicated as a cell, nematode, it can produce egg, then it will grow and reproduce. It's a life cycle, part of the life cycle. So finally, it's a growth and reproduction. Once it's growth and reproduce, then dissemination of the pathogens. So there are many pathogens, they are using several mechanisms to disseminate. Maybe air. If it is an air, we call it as an anemophily. Many of the rust pathogens, some of the fungal pathogens, they are airborne. Right? So while airborne, spores touch with the wet surfaces and get trapped, air movement stops, then it is washed out. For example, so for that's what I told you in this now dew season. The dew season, if you are going outside, sometimes will be sneezing. Right? Sneezing. Why? So because of this fungal spores will be there, pollens will be there. It is going inside through the respiratory tract. Your body is feeling that it's an antigen, then you react. Therefore, so many of these fungal spores, it need a wet to survive. Light moisture. Once it is touched, if the thin film of water is present in any host surfaces, the spores, if it is landed on that surfaces, then it can initiate the infection. If not, if it is sudden rain, it can wash out. So it will reach the soil, then there is no problem for your crop. right? So many of the fungal spores may be in the air, rain. That's what the people will say. It's a small rain is dangerous. It's a shower and cats and dogs are no problem. Precipitation, if it is precipitating, heavy rain, no problem. If it is slight showering, then there will be a problem, more chances for the fungal infections. Wind also helps the spreading of the spores. That's what it has been mentioned, that these fungal spores of the Basidiomycota rust fungi, it can move 1,000 kilometers. So I mean continent to continent. If it is one is airborne, if it is reached the air, can with the moving wind it can go 1,000 kilometers. This is equal to dissemination of the pathogen. Sometimes some viruses used for the dissemination, the insect vectors. So by the water also pathogen can spread. This is we call it as an entomo. So this is we call it as an hydrophily. So if it is in water, if it is by air, we call it as an anemophily. If it is by water, we call it as an hydrophily. If it is by insect, we call it as an entomophily. There are three mode pathogen can use to disseminate, right? So by the water, I mean from the moving irrigating water can spread your spores. That's what you know, if the initial symptoms is there somewhere in one spot of the field. So while irrigating, you are spreading that infection. The first step, if you're identifying any hotspot of the pathogen, first step is to stop the irrigation to that area, or passing the water through that area. If it is passing the irrigation, for example, in the fungal wilt diseases, mainly the irrigation is a very big problem. If you are irrigating the whole field, so in few days, your whole field will be infected. Okay? So these insect, especially nematode and the vectors, are sometimes transmitting this inoculum. That is we call it as an entomophilic, right? Dissemination of the pathogen. Some pathogens are seed-borne. Right? Some pathogens are seed-borne. So in the seed-borne pathogen means, so then from the planting material to planting material, it can transmit. Therefore, that's what before planting, you have to go for a good seed treatment. If not, you are transmitting that pathogen to the other. For example, CMUT fungi, blast fungi, Phyrecularia oryzae. So those are Helminthosporium oryzae, brown spot pathogen. So these are seed-borne pathogens. Sometimes human being also helping to transmit, especially the nematode eggs, like some of the fungal spores in the clothes. So one such example is Australia. Western Australia is free from rust fungi. Western Australia, Australia is a 4 region. So Western Australia is the western side. It's maybe 2,000, 3,000 kilometers from the other areas. So this is like an island-like continent. That was free from spores, rust. But suddenly one day they have observed that in the area with the spores. How it came? So because of the one farmer visited to US, the same cloth, he went to his field. Once it reached to Australia from US, the spores attached in the US, it came with him and it reached the field. It's created massive loss to the wheat. Right? So they have the human also helping. And also farm implements. Your tractor or the spraying implements, so mamati or fork, anything, rakes, anything can help to spread the pathogen. If you have used one area and not properly cleaning, sanitizing, you are using the other area. So spores, eggs, cells can transmit one area to other area. That is a problem in Sri Lanka. Many feel the human play a big role to transmit through these implements, right? So then this is the first disease cycle, but the detailed disease cycles and the examples are, I have prepared the handout. It is there in the LMS and I will open it, right? So you can read that one and get a more idea about, right? It's a disease cycle. It's a series of event. So it is starting with the inoculum. The inoculum may be primary inoculum or the secondary inoculum. The primary inoculum means, so from this produced spores or reproductive structure, starting initiation directly, we call it as a primary. The secondary inoculum is the inoculum produced by the primary infection, we call it as a secondary inoculum. The secondary inoculum starting the infection, we call it as a secondary infection. So if the inoculum is reached the host surfaces, then it will produce, it should stick with your host surfaces. This host surfaces, if it is stick for you, it needs some conditions. This condition means it needs thin film of water, mainly. If it is a thin the wet surfaces is there in your host surface, the leaves or stem, then the spores or the bacterial cells adhere on it. So for the adhesion, they are producing certain sticky substances by the spores. So then if the condition is suitable, the spore will produce, germinate, and germinating we call it as a germ tube. So if the germ tube touches the host surfaces, it produces structure. This structure, the broad structure, this is further helping to fix, act as a cushion or sucking pad. We call it as an aprosoria. These aprosorium can able to produce lot of degrading enzymes. It may be hemicellulose, cellulose, pectinase, bipectinases. So this aprosorium producing enzyme started to degrade the epidermis and wax layer and the cell wall of the host. So at the end of the aprosorium, it started to produce, it will produce a delicate sharp needles like structure we call it as a penetration peg. So this area is degraded. This penetration peg will give a pressure with the help of aprosoria. So how much pressure aprosoria can produce is a 1 car tire. How much of pressure is there in the hard tire? It may be 3.2 megapascal. Right? So that much of pressure, very small spores can produce. You can imagine that how much of pressure it can give. So pressure is like in car tire. Why? So it should break the cell. Right? So once the penetration peg is penetrated, so that at the time this host will recognize. So there are some of the foreign bodies. If the host is reacting, there is no any infection. If it is not reacting, or the defense is overwhelmed by the invading pathogen by producing antiweapons, we will study all these things later. Right? Antiweapons. Then pathogen can successfully infected. Right? Successfully infected. There is no any barrier. This is point we call as an infection. If it is infected, then these germ tube, the penetration peg, widen and start to produce an invading. It means going further and further and further. Invading mycelia. This invading mycelia may be intercellular mycelium in between the cells or through the cells. Intracellular mycelium or intercellular mycelium. Intercellular mycelium means going through. Intercellular in between. Intercellular mycelium. So if it is invading intercellular mycelium and intracellular mycelium, producing a structure to draw the nutrient, we call it as a hostoria. Right? Hostoria. This hostoria is drawing nutrition from the cells. Then fungal is more and more proliferate. If it is proliferated more, so then what will happen? So the pathogen will think that it's a good condition to reproduce. Will reproduce by producing spores and mycelia and the dormant mycelia structure. So if it is a nematode, it will produce eggs. If it is a bacteria, it will replicate, and viruses, it will replicate and produce new daughter cells. Replicate. So once it is fully reproduced, then it will disseminate. It should disseminate to other area. No? If it is disseminating and it is causing an infection in the same field, that is we call it as a primary infection. So the primary infection is producing another inoculum, we call it as a secondary inoculum. The secondary inoculum is producing infection in the same field, we call it as a secondary infection. That's the way the disease cycle is progressing. So normally, the primary inoculum only you can expect if the disease cycle is a once in a crop cycle. It's a monocycle. If it is a monocycle, many of the pathogens are monocyclic. So if it is a monocyclic, only one cycle per crop season. So therefore, you will not expect any secondary inoculum or secondary infections. But many of, some of the pathogens are monocyclic. Many of the pathogens are multicyclic. Within a crop season, it can complete 2 to 3 in their life cycles. At the time, you can expect the primary inoculum, primary infection, secondary inoculum, secondary infections. Okay, what it that is we call it as a disease cycle. The detailed informations are there in the handouts. Please. So up to now, you won't ask any questions, please. So next chapter is the effect of plant pathogen in plant physiological functions. So normally, so whatever if we are taking that pathogen means, so that is harmful to the host. So how it is harmful to the host means these pathogens are going to disturb the plant or the animal or any whatever the host physiological function. That is what it is detrimental. Otherwise, it is beneficial. So if that pathogen is causing dysfunction of the normal physiological function, that is we call it as a disease. How these cause defect or the interfering with the plant physiology, so we will discuss here. Right? Effect of plant pathogen in plant physiological functions. So there, you all know there are many pathogens. Here we are taking these three, and you can include if you want nematode. So nematode and fungi you can include here the same category because they are using the same weapons, right? Penetration peck by fungi, nematode by stylet, right? Stylet. So these four fungi groups, four pathogens you take it. What other physiological functions you would have studied in the plant physiology? You would have studied in the. I took that lectures too in the 1-2. Photosynthesis, respiration, permeability of the cell membrane, translocation of water and the nutrition, and transcription and translation. This is under the protein synthesis. Sometimes you would have studied in the agronomy the hormones, plant hormones and its functions. See, these are all, the respiration also we studied here. Those are plant physiological functions, right? If this pathogen is interfering with one of these functions, then the entire normal function will be affected, right? We will discuss how these pathogens are interfering with each and every. First is a photosynthesis. How they are interfering means, for example, if the invading pathogen is for invading pathogen to producing certain enzyme, I told you, they are to degrade these hosts. These enzymes, if it is degrading green content of the plant, like then you can observe the chlorosis of your leaf, necrosis, and reduce growth and yield. Right? So if it is created chlorosis and necrosis, it means green matter is less. The green matter is less mean your plant will fix the solar energy and it should not produce more amount of photosynthesis. That's what you will feel reduced growth and yield. And second is reduce the amount of photosynthetic area. Some of the powdery mildew and downy mildew, they are growing over the leaf surfaces. They are masking the leaf surfaces. So therefore, the green chlorophyll should not expose to the sunlight. There is no any photosynthesis. Means photosynthetic area is reduced. So therefore, your plant will show reduced growth and yield. And produce toxins. Certain toxins produced by the organism, these pathogens, they inhibit the enzyme or sometimes overworking of this enzyme. For example, if it's a nematode, they are producing certain toxic saliva, it's consisting more amount of enzymes. This enzyming mean as normal level, compared to the normal level, if the enzyme nematodes also producing the same amount of same hormone into the plant. So the if it is higher level, that's the plant, it will induce the plant to proliferate more and more. That's what you are expecting as a goal, right? Some of these, for example, MLOs, what will happen, and viruses inhibit the enzyme production, or it induce the level of progesterone more amount of, but certain other other than the auxin and cytokinin, they will produce certain the auxin and gibberellin, certain enzymes, the organism they are producing cytokinin and other enzymes. If it is producing or limiting that, then your plant leaves may be simone, its excessive branches of the will produce in your plant, or leaf. That's what you call it as a little leaf disease in brinjal and chili and everywhere, because of enzymes function is get destroyed. As stomata remain closed, unpartially closed, right? If the fungal producing mycelia is penetrating through the stomata, so the stomata opening is fully covered with mycelia. The opening is fully covered with mycelia, mean there is no any gas exchange. The hole is closed. Then how your plant will get carbon dioxide? So then there is no photosynthesis. Right? And also some of the organism targeting the gut cell of these stomata, especially viruses, then your stomata remain open. If it is remain open, what will happen? So during the hot season, hot weather condition, all the water present in the plant will get transferred. If it is get transferred, your plant will wilt and die. And photosynthesis will stop. If it is stomata remain closed, photosynthetic. In this way, these pathogens are interfering with the plant photosynthesis. These are some of these examples of the pathogen. For example, the viral infection, you see they are producing symptoms. These symptoms, what it is, how it is happening, right? They are reducing the here, there is no chlorophyll. If there is no chlorophyll content, so photosynthesis will be less. There is a photosynthesis will less means the plant will show the less growth, less reproduction. This is a virus infection. Then we will move to the other one. So the bacterial infection, you see, bacterial is causing necrosis. If it is necrosis, then suppression of the CO2 fixation because green matter content is declined. So if you are going there in these fungal infections, certain fungal infections, they are causing the blight. Chloroplast, RNA content, reduce the chloroplast number in RNA. And loss of chlorophyll here, chlorophyll lost. Then inhibit the electron transport chain, inhibit the photophosphorylation. So the fully photosynthetic photosynthesis is get disturbed. If we have forgotten the photosynthesis, how it is happening, please refer the 1-2. Please refer the 1. Respiration is arrested, and sometimes respiration will be increased, right? So respiration increases during the pathogen attack. It is as a research, it has been proved that one, especially if the plant is under the healthy condition, the respiration is normal rate. But in the infection started, plant produce more, plant is respiration rate is increased. So because it need more and more energy to defend. Especially the resistant varieties, respirations are higher, and susceptible varieties, respirations are less. So therefore, increased respiration depletes the plant reservoirs. The plant reservoirs is received, then your product yield of the plant will be small. Yield of the plant will be small. Sometimes it changes the metabolism. So effect of viruses in the hypersensitive host and non-hypersensitive, there are two hosts. If plant is having the resistance, right? Your plant is showing the very resistant. This is we call it as a hypersensitive host. If it is on, there is no any specific resistance, we call it as a non-hypersensitive host. If the normal, as an ordinary plant, so slightly increase the respiration. So in the hypersensitive plant, higher rate of respiration. This is research finding, right? Research finding. It's a bacteria. So the resistant tissues intermediate increase in oxygen consumption. So this is induced, it has been identified that so bacterial infected tissues are getting more amount of oxygen for respiration. So if it is getting more amount of oxygen, it means another way, plant is doing more respirations. Susceptible, don't reflect. This is also a research finding. For example, in the pepper with Xanthomonas versicolata, right? The Xanthomonas, they are causing the leaf blights, right? The vesicatoria, Xanthomonas vesicatoria is also producing, it showed that. It's a bacterial blights. And some of the fungi, the respiration is increased, the same disease plant, and it's normal respiration in the non-disease plants. So this is also increase in injuries created by the fungi, and decomposition of the tissues also increase the respiration, because if the plant is need to repair that lost tissues. That's what. So for respiration is increases. And membrane permeability is another factor. So membrane permeability means pathogens changes the permeability. So mechanical injury by the penetration peck or stylet, if it is created, then your plant permeability will get lost. So then your semi-permeable membrane will not work. So therefore, it's allowed to absorb all the things. Or sometimes your permeability is lost, it will not allow to absorb anything. So this is first detectable responses of the cells. Membrane permeability losses, you can expect the initial losses. For example, the fungi producing aprosoria fungi, the aprosorium of the fungi producing so much amount of enzymes to break the cell wall and the cell membrane. If it is broken, right, this is creating big injury in that area. Through that, plant inner content will be leaking. That's what in some plants, if you are making injury, you can find the gummy-like substances coming out because you have broken now. Broken the vascular tissues. Like that, here also your plant will get lost. Electrophoresis due to the leakage. Electrolytes, then your plant will weaken more and more. Sometimes some plant will die due to the loss of membrane permeability. Right, affecting function of the root. If it's a root pathogen, so if it is root hairs is get infected, so if it is root hair is damaged or rotten, absorb less water. That's what your plant is showing wilt symptoms. For example, in the fusarium and verticillium, or in the nematode attack, your plants will show the wilt symptoms in the day. It will become normal in the night. Because if the transpiration time is high in the daytime, it cannot absorb more because the root hairs get damaged by this pathogen. An effect of the viruses, right? So leads to the reduction in transpiration rate. I told you, so if the virus is infected, sometimes the stomata remain closed. There is no transpiration. And also reduce the leaf stomata aperture. Stomata aperture is reduced, so they cannot open it. Then there is photosynthesis is also affected. Transpiration is also affected. You see, both functions are affected. If it is created injury in the phloem tissues, the xylem tissues, you see the gummy-like substances are leaking out of these plants. Some like substances are leaking out of this plant. Effect of bacteria, you see, this is a fusarium and xanthomonas. This is Ralstonia. Ralstonia solanacearum and Pseudomonas solanacearum as a same. Tomato. They are causing the wilt. We call it as a southern wilt. You see the bacteria cells. If you are taking that, you can find it's this is bacteria, right? This is bacteria breaking and taking out bacteria. Here is the vascular tissues. Here, Xanthomonas in banana. You see, so leaking of the substances. So because why the silent tissues is get wound or blocked, and the effect of fungi. You see, this is one is a Fusarium oxysporum forma speciale cubensis in banana. We call it as a banana wilt. Banana wilt in banana. Banana wilt in banana caused by Fusarium oxysporum forma specialis cubensis. This is also the vascular wilt. It's a very big problem. Nowadays there are 4 strains. There are TR4. You would have seen that type in the website. You can find it how much of lost. So nearly $2 billion lost annually. Nearly 16 million hectares, especially in the Southern America, Southern America, not Northern America, Southern America, Ivory Coast, Brazil, and those banana producing areas, Costa Rica, Argentina, these 8 countries. So because they are the Cavendish is a major banana cultivating areas, and in Australia also some of the part is highly problem because of this fusing everywhere. And here you can say the effect of translocations. That's what I told you earlier too. What inorganic nutrient, because if the vascular tissues and the root chiasis get damaged, so this your plant cannot get normal, cannot do the normal function of the translocation, food translocation, water translocation, absorption, and transpiration. The whole function will be get affected. The root is get infected, those xylem tissues will get infected. Right? Get infected. So xylem destroyed. Some of the pathogen, for example, hanker. So xylem is clogged or destroyed. Then leakage and other things will happen. Loss of electrolysis, loss of vigor of your plant. That's what your plant will get break at that point. And some of the obligate fungal parasites, respiration increases, so photosynthetic product accumulates. So this because of the translocation. So the translocation is affected. Your photosynthesis, your synthesized sugars will be remain in the leaf. It will not translocate to some other areas. So this what will happen to respiration rate will increase because it should exhaust. Plant should utilize this one. Photosynthesis decreases. Why? Because stored synthesis material is remain there in the leaf. Then the leaf it will the plant will tell to leaf don't do the photosynthesis. It is there. So that is a problem. If it is EC, so many problems can happen. So many physiological problem happens due to this pathogens attack. So death of the pollen. If the viral attack, death of the pollen. That's how you can see that branch by branch dying of your citrus plants. We call it as a dieback in citrus. The virus disease. Citrus virus is causing the dieback. And transcription and translation. So transcription and translation, this is a photosynthesis, right? It's a part of the photosynthesis. You know, in the photosynthesis, DNA, mRNA will be formed from the double strand will be a single strand. It's a reverse transcription is happening. So for the protein synthesis, so coding will get disturbed. AGCT, AGC, right? This coding, 3 and mRNA, right? 3 nucleotides. mRNA coding is a process of photosynthesis, protein synthesis. So by the ribosome, right? If it is protein synthesis is getting affected because the coding will be problematic if it is viral infected. That is a photosynthesis will be get destroyed. And also changes in composition, structure, and functions of the chromatin on the DNA. That's what I told you. Coding. So we get normally will not plant produce as usual protein. It will produce some other proteins. So if it is some other proteins is produced, the plant defense will be activated because this is a protein also foreign body. No? increase activity of the enzyme and breakdown of the RNA. Enzyme activity will increase or breakdown of the RNA because of these pathogens at sometimes produce RNAs, enzymes. So RNA get broken. So there is no photosynthesis, protein synthesis. The high energy need for the translation. So sometimes it needs high energy. So if the increased production of the phenolics because of this pathogen attack for the plant defending, it produce more amount of phenolics. So if it is more amount of phenolics for the production of more amount of phenolics, it needs high energy. Right? So the resistant plants protein synthesis increase. The evidence is showing that protein synthesis is increasing if the pathogen is attacking. Why? So for the defense, a lot of defense material are protein substances. So therefore, it induces a protein synthesis. First few minutes of infection. Thereafter, it will become normal if it is a if it is a resistant plant. If it is a weaker plant, protein synthesis will be increased. Thereafter, it suddenly decline because pathogen overwhelmed. So these are the plant physiological functions. These plant physiological function, one or more of these combination of the plant physiological function is affected at the same time if the pathogen is attacking. That is a problem. If you are sick, if you are going to the doctor, doctor will ask if you have fever. It is indicating that something is there. So doctor will ask for blood checking, urine testing, fecal testing, an X-ray and everything. Why? So because if it is a one particle is there, many system will get collapsed. That's how the plant physiological function is the same. So doctors are studying in a human being, you are studying plant in agriculture biology. In animal science, you are studying animal diseases, poultry diseases, pig diseases. That is the problem. When in the entomology sites also you can studying in insect diseases. That's the thing. The beauty of the agriculturist mean you are the super scientist. Mean all the organism systems are you are studying except human being. Even digestive tract or enzymes or circulatory system and everything, all the system of these animals and plants you are studying except human being. That is the problem, right? That's a problem. So this is summary of this, how this plant, since the plant physiological function is getting affected. So mode of attack, the plant pathogen, we will study next class. So until now, you have any questions, you can ask